Hello, my today's topic is plant water relations. So under this topic, we will study about the water potential, psi W, solute potential, psi S, and the pressure potential, psi P, and DPD, that is diffusion pressure deficit. Then after that, we will study about imbibition and the plasmolysis. So what is water potential? So water potential is basically the free energy and the chemical potential of water. So for the pure water, the water potential is zero because the pure water has maximum capacity to flow, and if we add any solute into the pure water, then the water potential will decrease and it will not flow easily. So pure water has the maximum water potential of zero and it is maximum. And for the solution means if we add something so like any solute in the water, then in that case our water potential will be negative. So the psi W, that is water potential, decrease on addition of solute. Okay, so water moves from the high water potential to the low water potential. High water potential, that is there is only pure water and a little amount of solute is in it, so it will flow easily towards the container or the beak, towards the point where the water potential is very low, or in that case the solute is very high in the water. So if I talk about the units, then the unit is Pascal, or there is a question in the NEET that is 1 megapascal is equal to 10 bars. Learn it as it is. So now there is one term called solute potential. What is solute potential? Suppose it is a deficit or decrease in the water potential of a solution or water by addition of solute. So if I add, this is my one beaker and this is another one. Here the water potential is minus 10 pascal, and in this beaker I add suppose 10 gram of sugar. So in this case the water potential becomes minus 14 pascal. So from the minus 10 pascal to minus 14 pascal, there is change of minus 4. So this deficit or decrease in the water potential due to addition of solute is called solute potential. At normal atmospheric pressure, psi W is equal to psi S. Okay, and the psi S, that is solute potential, is never positive, it is always negative. We need to remember this. And there is one statement in NCERT: if pressure greater than the atmospheric pressure is applied to a pure water or the solution, its water potential will increase. You can all easily understand it. If suppose there is a container, we need to flow water from here to here. If we apply some pressure over here, then it will easily flow. Water will easily flow from here to some other medium. So if the pressure greater than atmospheric pressure is applied to the pure water or solution, the water potential will increase and it will flow easily. So maximum water potential is, I have told you, that is 0 of pure water, and when we add solute into the water, then water potential will decrease. We have learned this till now. And there is one more term, that is pressure potential. So what is pressure potential? Pressure produced in cell or plant system due to presence of water. मतलब ड्यू टू प्रेजेंस ऑफ वाटर इन अ सेल में जो वाटर है उसकी वजह से जो पोटेंशियल जनरेट हो, जो प्रेशर बन रहा है सेल में, दैट इज प्रेशर पोटेंशियल. सो प्रेशर पोटेंशियल में बी नेगेटिव एंड इट मे बी पॉजिटिव. ओके, सो by combining all this, water potential is equal to solute potential plus pressure potential. So till now we have discussed about water potential, that is maximum 0 of pure water. When we add solute into it, it will become negative. Okay, then water will move from high water potential to low water potential. Solute potential is decrease in the water potential of solution, water by addition of solute. So here from minus 10 to when we add sugar solute into the water, then the water potential will decrease from minus 10 to minus 14, and the decrease in the water potential is solute potential, that is minus 4 pascal. At atmospheric pressure, water potential is equal to solute potential. Then the pressure potential is the pressure produced into the cell, in the cell, due to presence of water. It may be positive or negative, and this is the combined formula. I will discuss more about it when I will discuss diffusion pressure deficit in the end of the lecture. Before that, I'm gonna tell about osmotic pressure. So what is osmotic pressure? Before understanding it, let us discuss an experiment, that is Thistle funnel experiment. Okay, so this is the diagram of thistle funnel experiment. This green one is the semi-permeable membrane. Okay, and this is the water. So the water will move from this beaker via the semi-permeable membrane to this flask. Okay, so to prevent this osmosis, this is osmosis, the movement of water from semi-permeable membrane from high water potential to low water potential. This is osmosis, you know already. So to prevent this osmosis, we apply a pressure from above. This pressure is osmotic pressure. Okay, so this osmotic pressure is to prevent the osmosis, and it is always positive. We are applying this pressure over the flask, and it is applicable in the closed system. Okay, then there is a term that this was osmotic pressure. There is one more term called osmotic potential, or we can say solute potential. We have already discussed that solute potential is always negative. Okay, so this solute potential is also known as osmotic potential, and it is applicable in the open as well as closed system. From here we come to know about a term called, there are two terms, similar term, osmotic pressure and osmotic potential. Osmotic pressure is negative of osmotic potential, or we can say osmotic pressure is minus Is, that is solute potential. For example, if I say that is osmotic pressure is suppose plus 60 pascal, then what will be the solute potential? It will be, it's negative, that is minus 60 pascal. And if psi s is suppose minus 10 pascal, then osmotic pressure is, it will be plus 10 pascal. So now we will understand a term called plasmolysis. It was a little bit concept about osmotic pressure and osmotic potential. Now plasmolysis. So to understand this plasmolysis, we will see it by the diagram. So plasmolysis is basically the shrinkage and withdrawal of protoplast from cell wall if plant cell is placed in hypotonic solution. So I think you didn't get what I was saying. So we will see from here. I have drawn this cell. This is the cell wall, white valley. This is the cell membrane, and this is vacuole. Okay, this is our normal cell. Okay, so when I place this cell in the hypertonic solution, you can see the vacuole size will be reduced. So I will write the vacuole size is reduced. Okay, and Turgor pressure is 0. I will discuss it later. And the protoplast is in the contact with cell wall. There is no like withdrawal of the protoplast. The protoplast is in contact with the, this is cell wall, cell membrane, and protoplast. It is in the contact with cell wall. Okay, now in next case, there are basically 3 steps of plasmolysis. We will discuss all of these. The one I have discussed is the limiting plasmolysis, then is the incipient plasmolysis, and then is evident plasmolysis. We can learn it by a mnemonic called lie. Okay, so in the next stage, this is our plasma membrane. The plasma membrane will be like this. What you can see here is the protoplast first contract from the corner. From here the protoplast first contracts. And the Turgor pressure is negative. I will discuss it after some time. And if I talk about the vacuole, will decrease in size. Okay, in the next case, we can see the protoplast become almost round like this, and the vacuole will be small in size. Okay, so the protoplast becomes round and the Turgor pressure will be more negative. So what is Turgor pressure? I am saying it again and again. So this is the cell wall. Okay, and this is the cell membrane of a cell. So the cell wall applies a pressure in this direction. This is known as wall pressure, and against, in opposition to this pressure, the cell membrane applies a pressure called Turgor pressure. Okay, so it is a Turgor pressure. So in the plasmolyzed or in the flaccid cell, here the Turgor pressure will be zero. Both the pressure, the Turgor pressure will be zero, and here the cell, the protoplast has contracted from the corner, so it means the Turgor pressure would be less than the wall pressure, so that the wall has been contracted now, so the Turgor pressure is negative. And in this case you can see the protoplast has been contracted from all the sides. If it is contracted from all the side, it means that the Turgor pressure would have been much more less than the wall pressure. So in this case the Turgor pressure is more negative. Okay, so I have discussed about the plasmolysis, the three stages. You can remember it by mnemonic like lie, limiting, incipient and evident plasmolysis. You need to understand about the vacuole size and the Turgor pressure and the protoplast condition. Okay, so what is plasmolysis? The definition of it is a shrinkage and withdrawal of protoplast. Okay, in the steps I have said about the protoplast, the shrinkage and the withdrawal of the protoplast from this one cell wall if plant cell is placed in the hypertonic solution. Okay, so now this plasmolysis is a reversible process. What it means that if we place this plasmolyzed cell in a hypertonic solution, then the whole process will be reversed, and that is known as deplasmolysis. So what is deplasmolysis? The plasmolyzed cell regains its original shape original state if it is placed in the hypotonic solution. If placed in hypotonic solution. Okay, so I have discussed the stages of plasmolysis. There are three stages. Number one, limiting plasmolysis. Number two, incipient, and number three is evident. Okay, so I will tell you two points. In the plasmolyzed cell, the Turgor pressure is negative. I have told you why Turgor pressure is negative, because the protoplast has been contracted from cell wall, either from the corners and in the next step from all around. So the protoplast will become round in shape. And in the flaccid cell, the Turgor pressure is zero. We will see this when we will discuss about the diffusion pressure deficit in the subsequent part. So there are two uses of plasmolysis. Number one is what? The bacteria. Bacteria are killed in the high concentration of salt and water. Very easy. Also the weed killing in the farms by sprinkling of salt. It is in hypertonic solution. Salt is in hypertonic solution. So by sprinkling the salts over the weeds, there will be the plasmolysis of the cells and the weeds will be killed. Okay, so there is one more term called imbibition. So what is that? It is the absorption of water by hydrophilic solid substances without forming a solution. So it is absorption of water, that is imbibate, by hydrophilic, by what? By hydrophilic water loving solid substance, that is imbibant, solid substance without forming a solution. Okay, So it is without forming solution. So have you ever seen that in the rainy season the woody doors, they are very difficult to close if they come in contact with the water in the rainy season. If you have not, like uh, there is one application that is the swelling of wooded doors in the rainy season. I have seen it clearly in my day-to-day life. In the rainy season, the door will be difficult to close. Why? Because it absorbs the water from the it absorbs the rain water, and then it will increase in the size, and it will not fit into the this cover of the door, and it will be difficult to close. So it is a very good example of imbibition. Another one is the seed germination, and another one is the breaking of rock by wood by prehistoric men. The one what they did used to do was, in the rock, if they want to break any rock, suppose this is my rock, okay, they used to put the wood inside that rock, and then wood will, what will it do, this wood will absorb the water from the rock. Okay, so imbibition will take place here, and there will be the cracks into the rock due to loss of water, so the rock will be broken down. It is very good example. Okay, so it is basically application why we are studying this imbibition. And so what is that, I have told ki absorption of water by hydrophilic substance like the wood without forming a solution. So in this case the solid is known as imbibant. I have already told solid is known as imbibant or absorbent, and the liquid, that is the water which is being absorbed, is called imbibate. Okay, so when they will get absorbed, if the solid and the liquid both would have the both should have high affinity. So these both should have high affinity for this imbibition to take place. So there are this is one thing that is commonly asked in paper. It is not given in NCERT, I think, but it is asked in the NEET. It was asked in NEET. So good imbibant are, what are the good imbibant, the good solid thing that will absorb the water are the phycocolloids. Number one, it is good imbibant, very good. Then pectin, less than that, protein, starch, and the cellulose. You need to remember this. Okay, so there is one thing called imbibition pressure, or we can say matrix potential. So phycocolloids having the highest imbibition pressure, and that will decrease subsequently till the cellulose. Okay, I will discuss it later. So what are the characteristics of this imbibition? The volume of imbibant will increase. Of course the volume of imbibant will increase. That's why our door was not closing na in the rainy season. So, and another one is, it is reversible process. So the door that was not being closed in the rainy season because of absorption of water. So when there is a sunny day, then the water will be lost from that door, and it will be easily closed, okay, after 2 to 4 days. So it is a reversible process. Another effect is that it is an exothermic process. Okay, the pressure produced during the imbibition is called the imbibition pressure. I have told that here, or the metric potential. So application I have already discussed. One more thing I will discuss is the diffusion pressure deficit. It is very important topic. Okay, so what is diffusion pressure deficit? So basically DPD, diffusion pressure deficit, or the suction pressure, indicates the demand of water. Simple thing, it indicates what? Demand of water. So what is the definition? Decrease in the diffusion pressure of water, decrease in diffusion pressure of water in any system by addition of solute. So if we add any solute into the water, it will become a solution. There will be the decrease in its diffusion, there will be the decrease in its flow. So that is the diffusion pressure deficit. So what it indicates is that we need to add some water into the solution so that it will become less dense and it can easily flow. So diffusion pressure deficit, it indicates demand of water. So diffusion pressure deficit is always negative of water potential. So if water potential is generally 0 in case of the pure water, and it is negative generally in the solution. So if diffusion pressure deficit is negative water potential, then it would be surely always positive, na? So it is always positive. One more thing I have, this is our, let me tell you, the water will flow from the low diffusion pressure deficit to high diffusion pressure deficit. Easily understandable. Next one is the diffusion pressure deficit is directly proportional to the solute concentration. That is, it depends on the how much solute is present in the water. Now, if more solute is present, then there will be the demand of more water. Okay. So there are some equations we need to discuss. That is, diffusion pressure deficit is equal to osmotic pressure minus Turgor pressure. How we can prove this equation? Let us see. I will do it once more. We know that is the water potential is equal to solute potential plus pressure potential. We know that. Okay. So solute potential is negative of osmotic pressure. Okay, I have told you when I was discussing the difference between osmotic pressure and the osmotic potential. Okay. So another thing is the psi p, pressure potential, is equal to the Turgor pressure in the cell. You know these two things. So by putting these things in our this equation, so psi w, that is water potential, will be Turgor pressure minus osmotic pressure. Okay, and we know one thing, that diffusion pressure deficit is equal to minus psi w. Okay, so if I put this diffusion pressure deficit over here, I need to apply this minus sign over this side now. So our diffusion pressure deficit is equal to osmotic pressure minus Turgor pressure. Very easy. So now we will see some examples of this equation. So it is our in the normal cell, in the fully turgid cell. If our cell is fully turgid, it means what it means, कि the wall pressure and the Turgor pressure both are equal in this case. So the Turgor pressure is equal to the osmotic pressure, and if this both things are equal, then they will cancel each other. So diffusion pressure deficit in the turgid cell is 0. We can practically see if the cell is already so turgid, then there will be no demand of water in that cell, right? Okay. If the cell is flaccid cell means Turgor pressure is 0. Okay, Turgor pressure is 0. So diffusion pressure deficit is equal to osmotic pressure. Okay. So in the plasmolyzed cell, we have already discussed there is the contraction of protoplast from the corners, and then the protoplast will become round in the last evident stage of plasmolysis. So the Turgor pressure in that case was negative. Okay, so diffusion pressure deficit would be OP minus TP. If TP is already negative, so the diffusion pressure deficit would be OP plus TP. Easy. So what will be the order of diffusion pressure deficit? In the case 4, it will be the maximum. Then in the third case, then in the obviously first case, and in the second case it is already 0. So this is all about my today's topic. If I summarize the topic, so we have discussed about the psi w, psi s, psi p and dpd, their relations and their equations in the normal cell, plasmolyzed cell, turgid cell and the flaccid cell. It is very important. You can expect any 1 to 2 questions from this topic in the NEET. So imbibition I have told you, and the plasmolysis. Nothing to remember in the water potential. It is basically the free energy of water to move. Pure water may, it is 0. For solution, if we add solute into it, it will become negative. The water move from high water potential to lower water potential. Unit is Pascal. Solute potential is a decrease in the water potential of the solution by addition of water, by addition of solute. You can easily understand it by this example. Okay, this is very important. At atmospheric pressure, psi w is equal to psi s. Psi s is never positive. Okay, and this is very common term, common line in the NCERT. I have told you pressure potential is the pressure produced in the cell due to presence of or entry of the water. It is positive or negative. This is very common equation and very easy. Okay, osmotic pressure and osmotic potential I have told you. Osmotic pressure is the pressure to prevent the osmosis. Osmotic potential is the same as solute potential, and osmotic pressure is so minus of solute potential. Then plasmolysis, you can understand by these three diagrams. There are three stages: LIE, limiting, incipient, and the evident. There is the contraction of in the first stage, vacuole will reduce in size, transfer pressure will become zero, and the protoplast is in the contact with cell. Second stage में protoplast contact from the corner. Third stage, protoplast will become negative and the transfer pressure will subsequently be more negative. Okay, so definition I have told you, it is a reversible process. So there is a term plasmolysis if we place the plasmolysed cell in the hypotonic solution. Stages I have told you, and the transfer pressure in the plasmolysed is negative I have told, and flaccid में 0. Application are varies in the weed killing and the bacteria killing. Imbibition is the absorption of water by the solid. Okay, you can remember this imbibition by your day-to-day life, like example of the wood in the rainy season, swelling of the woody doors in the rainy season, it is very good example of that. And the breaking of rock by the wood, it is also very good example. Okay, so reversible hai, exothermic process, and the volume of imbibement will be increased. Okay, then there is the diffusion pressure deficit. General line for that is that it indicates demand of water, negative of psi w. There is one equation you need to remember, or you can easily prove that also if you have basic idea of all the pressures and the potentials. So dpd is equal to op minus tp, and for this further these cells you can remember this equation. This is very important. Thank you.